Hello, hi. Welcome to the third part of the lecture series. Today, in this lecture, we will be dealing with the dihybrid cross and also the law of independent assortment. Here we know that in the previous lecture, we have dealt with the monohybrid cross and also the law of dominance, law of segregation, and also the multiple allelism. Multiple allelism means more than two allele present in a population. Okay, we know that in a diploid organism only two allele will be present, alternative allele will be present, only two allele will be present in a diploid condition. But in a population we know that more than two allele can exist. So these were the things we studied in the last part of our lecture. In this lecture we will be dealing with the dihybrid cross and also the law of independent assortment. So what was monohybrid cross? In monohybrid cross we took only one particular character at a time. We were only considering a particular character at a time. Like for in Mendel's experiment there were seven contrasting character of a plant, a pea plant, right? So in monohybrid cross he only took one particular character, which means we have done the cross, right? We have done the experiment with height. We can done with other characters also. Here in dihybrid cross we will be checking, or we will be doing the cross with two character taken at a time. Okay, so first today we will be dealing with the dihybrid cross, right? So dihybrid cross, a hybrid is formed by taking the two character. Hybrid is formed by taking two particular character at a time. Okay, we are only focusing on two different characters. There may be other character present, but here we are only focusing on two different character. So I have said that in a pea plant Mendel took about seven contrasting character. Mendel, because of that seven contrasting character present in a pea plant, he was able to take the pea plant as an experimenting organism, right? So and he was able to cross and make the next generation very easily because of the pea plant. And also in pea plant the seven contrasting character were seen very easily, right? The height, the color, or the shape of the pod, or the seed color, seed shape, etc. were very easily noticeable. So for this reason he took the pea plant, right? So in dihybrid cross I am taking so also we have dealt about the results from the monohybrid cross. What were the results from the monohybrid cross? We know that phenotypic ratio and genotypic ratio, right? Phenotypic ratio, we know that phenotypic ratio 1:2:1, right? And also phenotypic ratio 3:1. 3:1 means 3 tall and 1 dwarf, and phenotypic ratio means 1:2:1. We know that 1:2, which means the genotype was TT and small t. These were the genotypes, right? So it will be formed one number of this genotype was 1, number of this genotype was 2. So 1:2:1 was the genotypic ratio of the Mendel's monohybrid cross. Also we know that the phenotypic ratio was 3:1, 3 tall and 1 dwarf, right? Yes, 3 tall and 1 dwarf. This was the condition in monohybrid cross. So let us check what will happen in dihybrid cross, right? So we can check it right now. So we are going to cross the dihybrid. We are going to do the experiment, right? So we are taking two different character at the same time. So out of 7 character we can take any 2 character. I am taking the endosperm color. Okay, the endosperm color and the seed shape, which means the seed color and seed shape. Okay, seed color does not means seed coat color. I am taking the seed endosperm color. Actually the 7 contrasting character, the seed color is, which means the seed coat color, seed endosperm color. Okay, so I am taking two different character. First character is the seed color. Second character is the seed shape. Okay, we know that seed color and seed shape. If I am taking the seed color, we know that there are two different color for a pea plant. Also seed shape, there are two different shape, right? Seed color, we know that seed color can be yellow or green, and the yellow is the dominant form. It is the recessive form. And also seed shape color. Seed shape color can be rounded. Seed shape can be round or wrinkled. So I can write it. Seed shape means round or wrinkled. Round or wrinkled. Round is a dominant character. Wrinkled is a recessive character. It means in presence of a dominant allele of round, the wrinkled form will be masked. In presence of a round color, in presence of round shape, which means dominant form of round, which is either capital or capital small, right? Homozygous or heterozygous. So in this condition, the wrinkled form will be masked by the dominance of the round. This is what we have discussed in the principle of dominance. Okay, this is what Mendel concluded or Mendel understood from his experiment, right? The masking effect of the dominant gene, dominant allele, right? The gene is same, but the allele, due to its heterozygous or homozygous. So here we are taking two genes, the gene for shape and gene for color. So two different genes, that means two different character are taken at a particular time or a particular cross. Okay, so this is the general idea about the experiment. So we are going to do the experiment. So first I will wrap this off. I hope if you write it with me in your notebook, it will be very useful for you afterwards. So I'm going to do the cross. So the parent generation, parent P1, P2. Let P1 be male and P2 be female. Actually in Mendel's cross there is no need to write male as a parent 1 or female as a parent 2, because even if we do it vice versa, the result will be same. I am saying this because here the result will be same, but in certain other crosses like cytoplasmic inheritance, the cross which we will be dealing with later. So in that case the reciprocal cross will not be same. Here the reciprocal cross will be same. We will find out how the result will be same, right? First we are going to cross. So we are crossing parent 1 with parent 2. Parent 1 is a male and parent 2 is a female. So I am taking parent 1 as a homozygous dominant. So we are taking two different character, seed shape and seed color. So I forgot to write that. See, for seed shape, round and wrinkled, round will be the dominant form, so capital R. Okay, and wrinkled will be the recessive form, so I will be writing small letter r. In seed color, we know that yellow and green, so I will be for dominant form capital Y, for recessive form small y. This is the condition. Okay, so this is the parental condition. So I will write the parent 1 will be homozygous dominant and the parent 2 will be homozygous recessive. Actually, you may, in your mind, you may have arise question that why he is taking homozygous dominant or why he is taking homozygous recessive, because this is the cross which Mendel have done to conclude his law of certain laws, right? So this we are studying the particular experiment. So this particular experiment should be in that particular order which Mendel have done. So for this reason we are studying in a particular order. For this reason we are taking parent 1 as homozygous dominant and parent 2 as homozygous recessive. Okay. So parent 1 homozygous dominant is a dominant. So capital R, capital R, capital Y, capital Y. So what will be its phenotype? Phenotype will be round yellow pea plant. Round yellow means round seed, yellow color seed, right? Pea plant. So here in parent 2, it is homozygous recessive. Y, Y. Okay. So it is wrinkled green pea plant. This is the condition. This is the parental condition. So we are going to do the cross. Okay. So we are going to make the gametes. How the gamete is formed, we know that gamete is formed by the process of meiosis, right? Reduction and division. Here the condition is 2n, right? We have studied the basics. Here the condition is 2n. 2n means it will be having two set of chromosome. One set of chromosome here, one set of chromosome here. One set of chromosome will be having the R and the other set of chromosome will be having this R. The same set will be having Y, the other set will be having Y. We have dealt very clearly in the first lecture, first part, right? So if anyone is having doubt, please go to the first part of this lecture, understand the basics and come back. Okay, so we know that this is in a haploid condition because a gene is having a two different allele here, right? Here it is in a homozygous condition. Here also it is in a homozygous condition, but it is in a recessive condition, right? Okay, this is parent 1, this is parent 2. Parent 1 will be wrinkled, having a wrinkled green pea plant. It will be having a round yellow pea plant. So after the process of meiosis, the gametes will be formed. Okay, so what will be the gametes here? Here the gametes will be, by the process of meiosis, the gamete will be RY. Gamete will be formed as RY. This is the gamete RY. Okay, here only one type of gamete will be formed because I earlier mentioned that here only we can calculate the type of gamete by an equation 2 raised to n. N is the number of heterozygosity. Here there is no heterozygosity, so 2 raised to 0. So 2 raised to 0, 1. Right? So here only one type of gamete will be formed. That type will be capital R, RY. Right? It is correct, right? After the process of meiosis, the gamete will be formed. We will discuss it later how the gametes are formed in cell division class, right? Actually, just understand this. After the process of meiosis, the gametes will be formed. Here the gamete is capital R, capital Y. So what will be the condition here? After the process of meiosis, small r, small y will be formed. This is the condition in meiosis, in the parent 2, which is the recessive form. So the gametes have been formed by the process of gametogenesis, right? Here also gamete will be formed by the process of gametogenesis. So it is a male plant, so it is a female plant, so the pollens will be transferred from this plant to this plant. So it is actually not needed, but we will just understand it. So the pollen is transferred and the process of fertilization happens here, and a zygote will be formed. Okay, so by fusion, the fusion of two gametes, right? This both will fuse and form capital R, small r, capital Y, small y. So this is the condition of the first generation, first progeny, first generation, filial generation. I earlier F1. So this is F1 generation. What do you mean by F? F means filial. Filial means daughter or son. It's actually a Latin word which means daughter or son. So first F1 generation formed will be capital R. This is the genotypic condition. So it is a diploid form, right? This allele will be having its alternative. This gene will be having both alternative forms, right? Both alternative alleles. So what will be the phenotype of this condition? What will be the phenotype of this progeny? The phenotype will be, we know that it is in a heterozygous form, right? So this capital R will mask the small r. So in this condition, it will be forming a round plant, right? A round seeded plant. In this condition, capital Y, and this is also a heterozygous form. So this capital Y will mask the effect of the small y. Small y can't express fully. It can't express. So capital Y will be expressing fully. So it will be having an yellow seed. So in the F1 generation, an round yellow pea plant will be formed. Right? Same as this condition, same as the parental generation. One of the parent, right? One of the parent has been expressed in the next generation. Every pea plant in the F1 generation will be having round yellow, so that its mother's phenotype is not expressed, only its father's phenotype is expressed in the F1 generation. So this is the first part of the Mendel's dihybrid cross. Now what we are going to do is that what Mendel have did is that he is going to cross F1 with other F1. Why did he cross F1 with other F1? Because in order to find why did his mother phenotype was not expressed, right? I have said it in the monohybrid cross also. Look, Mendel have did the cross. In the F1 generation, he was shocked that one of the parental generation was not able to see. Only one of the parent was being able to see. So for this reason, he have done, what did he do? He did that, he crossed F1 with the same F1, which means F1 has been self-crossed, self-fertilized. So we will do the next part right now. So F1. There is no need to write male or female here because the result will be same. The result will be same. I will show you why did the result was same in the reciprocal cross. So I am taking the genotype. So he self-fertilized, right? So RR selfing, RRYY, RRYY. This is the result of the progeny genotype. Okay, round yellow seeds. So we are going to fertilize. We are going to fuse it once more. So the F1 plant have been formed and seed have been formed. The plant have grown and it have grown to a certain stage that it can produce now pollen grains, and also its pistil is ready. Pistil is ready to accept the pollen grain, right? So we are going to, so they are ready. So now by the process of gametogenesis, pollen grains are formed, ovules are formed. So how the gametes are formed? By the process of meiosis, right? So in meiosis, the gametes will be formed. So here, how many type of gametes will be formed? You have to say, because I have said that type of gamete, we can find the type of gamete as 2 raised to n by giving the equation 2 raised to n. How this is formed? It is pure mathematics, right? So type of gamete is 2 raised to n. So here n means number of heterozygous gametes. So how many heterozygous gametes are here? 1, 2, right? 1, 2. So here 2 heterozygous gametes, which means n is equal to 2. So 2 raised to 2, which means 4. Which means 4. Which means that 4 types of gametes can be formed. Here also the condition is same because the genotype is same, right? Here also 2 raised to 2. 2 raised to 2 gametes. How the gametes is formed, I will shortly explain how 2 raised to 2 type of gametes can be formed. So what are the gametes, right? The gametes will be capital R, capital Y, capital R, small r, capital Y, and small r, small y. These are the 4 types of gametes. I am saying type, not the number, because a plant can produce several gametes in its lifespan. I am only talking about the type of gamete. Here also capital R, capital Y, capital R, small r, capital Y, small r, small y. These are the types of gametes. So here 4 gametes will be formed. Here also 4 types of gametes. 4 types of gametes will be formed. So if we fuse it, how many type of gametes will be formed? We know that 16 gametes will be formed, because if we take this particular gamete, it can fuse with this. So one gamete can fuse with 4. So 4 gametes can also fuse with 4. So 4 into 4 means 16 types of progeny can be seen in the F2 generation. 16 types of gametes can be formed in the F2 generation. In order to understand the genotype and the phenotype of the F2 generation, we need to draw a checkerboard, a Punnett square, right? So before drawing the Punnett square, I will explain how these gametes were formed by the process of meiosis. Actually, if we look at these four gametes, we can understand. If we look at these four gametes, we should understand something, that capital R and capital R capital Y and small y were the gametes of the parental generation. If you remember, in the first parental generation, if you are having capital R capital Y parent 1, small r small y. So these the two gametes marked here are the parental gametes. And what about the other two here? These are non-parental gametes, which are formed by the process of recombination. So here, in four gametes, two gametes are parental and two gametes are non-parental. This is by the process of recombination. So in order to understand that, we will now, before drawing the checkerboard means we are fusing, or we are fusing the gametes. Now the gametes are already formed, we are fusing the gametes to form the zygote. So before that, I will explain how these four types of gametes will be formed, right? Okay. Okay. Just consider a cell. Okay. Cell is having a chromosome. I am only drawing a chromatid, right? This is I'm only drawing a chromatid, which means we know that this is a chromosome. I am only drawing a part of the chromosome. So this part is this actually. Chromosome in its replicated phase, right? In S phase, we know that in synthesis phase, a chromosome will undergo replication to form a sister chromatid also, right? I have earlier explained how this is formed, right? So I think there is no need. So we are only taking this part. So after cell undergoing G1 S G2 phase, it will go directly to the meiosis phase, right? Meiosis or mitotic phase. Here we are dealing with the process of gametogenesis, or process of production of gametes. So here we will be dealing about the meiosis. Meiosis is the production, reductional division. So we are considering a cell at prophase. Considering a cell at prophase, metaphase, anaphase, telophase, and cytokinesis, right? So here I am considering a cell at a prophase. I think I should draw it very largely, right? It may be not visible in the board. I will draw it much more larger. A large cell, okay? So here we are going to understand the gametogenesis of the gamete formation or the meiosis in the F1 generation. F1 generation here have capital R, small r, capital Y and small y. So it is in a heterozygous condition. So we are going to understand the meiosis in the F1 progeny, F1 generation, right? So I am considering two homologous chromosomes. Let it be chromosome number 1, the same homologous chromosome, chromosome number 1, chromosome 1, chromosome 1. Okay. Now here I will write chromosome 2, chromosome 2. I actually earlier mentioned what is homologous chromosome, right? So this is chromosome number 1, chromosome, the same homologous chromosome pair, right? So I am writing here we are considering about the F1 generation, capital R, small r, capital Y, small y. We are going to find how the gametes, 2 raised to 2 gametes were formed. Why the types of the gametes arise like this, right? So I'm going to write the genes here. So actually there are 2 genes What were the genes? Capital R gene and Y gene, right? R gene which codes for the shape and Y gene which codes for the color. So R. Just I am taking any part of the chromosome. Just consider the R gene is present here, capital R. So we know that the same capital R in the same will be present at the same locus, right? That is why this is called as alleles, because they are present in the same locus. So we have drawn one here. In the next homologous chromosome, it is having its alternative form, which is the small r. So it will be having the same r in the same position. So alleles. Okay. How these were formed? How the pair of how the sister chromatids were formed? They are formed in the S phase of the replication. These are the basic understanding, right? We have studied from 10th, right? I think so. You should have a better understanding about the basics of the meiosis. Okay. So in chromosome 2, just imagine that the second gene is present in the any other chromosome. I am just considering the second gene, that is the gene which codes for the color of the seed, is present in chromosome number 2. So I am just considering gene Y which is present anywhere here. So the same will be present here, and its alternative form small y will be present here. Look, this is the condition. Okay. So this is the possibility number 1. Why did I say this is possibility number 1? Because there is another possibility of the arrangement of the chromosome here also. What is the next chance? I will draw the next chance here. So the next possibility, which is the possibility number 2, this possibility can lead to the recombinance. How? I will show right now. Chromosome number 1. Chromosome number, homologous chromosome number 2. Okay. So I am here writing capital R, small y and small y. Chromosome 1. Chromosome 1. So the next possibility is that the gene will be in the chromosome. The alternative form of this gene will be present, which is the recessive form may be present here. So there arises two different possibilities, even though the possibility is different, but their genotype will be same, right? Capital R, small r, capital Y and small y. So just look the condition here. We can also change this also, but there is no use, right? It will be same. We can write the capital R, small r here also, but it will be same as the previous one. So there are only two different chances. The chances here are 50% it can occur and 50% it can occur. It is because how this occur, it is according to the orientation of the chromosome. Chance, possibility that capital Y will be present with the orientation will be like this. Capital R dominant here. The chance may be here dominant may be present of the second gene, or the recessive form will be also present in the second gene in here. So the condition can vary. It is according to the orientation of the chromosome. So what will occur here? There is lack of space. So what will occur here in the cell division phase? What will occur in the meiosis 1? Meiosis 1 means the segregation of the homologous chromosome. Segregation of homologous chromosome means this homologous chromosome, this chromosome will go to, I will draw a line here. This homologous chromosome number 1 and chromosome number 2 will go to the polar side, and this will also go to this polar side. So the cell will divide in the meiosis 1. So here what will be formed? I am just drawing because of the lack of space. Here what will be formed? Capital R, and capital Y, capital Y. Here what will be formed? Small r, small y, small y. So this is the possible condition, right? First possibility is, there are actually two possibilities. The possibility one is that, actually this is known as cis form, and that is the transform, or it is known as the repulsive form. Actually that is actually not needed, but I am saying it for your information. So in this possibility number 1, the gametes can be formed which are that of the parental condition, right? So this is after meiosis 1. So meiosis 1, in the anaphase, the segregation of the homologous chromosome occur, which means chromosome 1, it can segregate, it can separate from each other, right? So chromosomal segregation can occur. So chromosomal segregation can occur. So this is after the result of meiosis 1. Now what happened after meiosis 2? After meiosis 2, we know that segregation of the sister chromatids. Meiosis 1 anaphase will form segregation of homologous chromosome, and in meiosis 2 it will form the segregation of the sister chromatids. So we know that this is a sister chromatid, this is a sister chromatid. They will separate. Okay, they will separate and form RY, and also RY, right? So after further division, they will be forming cells, right? This is cell, this is cell, and a cell. So here 4 cells will be, cells will be formed, and you see the condition is equal to that of the parental condition, which means the parental genotype, right? So this is the possibility of the possibility in which parental combination can be formed, and there is also a 50% chance that if the chromosome are in this position, which means if the chromosome are maintained in this condition, then what gametes will be formed? So here after meiosis 1, capital R, small y, small r, capital Y. After meiosis 2, what will happen? Capital R, small y, capital R, small y, small r, capital Y, small r, and capital Y. So this, here 2 types of gamete are formed. Here also 2 type of gametes are formed. In this 2 type of gamete, it will be in a recombinant manner, which will be which is a non-parental manner. So this is the condition which leads to the 4 types of gamete. This is the thing which I have mentioned. But you know, Mendel was not aware about this chromosomal segregation, or he was not aware about what is happening in the cell, but he just concluded his result according to the Mendel cross. The results which he got from the dihybrid cross and monohybrid cross and certain other crosses also. So but after like 100 years later, the actual process was found out by, and the chromosomal theory of inheritance was studied, which means the name factor which Mendel mentioned factor, now they called it as a gene by genetics, or a Danish breeder. He named it as a genes, right? William Johansen, right? So he named it as a genes, which means the factor which are transformed from the parents to the next generation. So this is the condition of the possibility of the parental and non-parental type, and it is the basis of the chromosomal basis of inheritance, which means actually the inheritance is occurring in the chromosome. We will study in certain other cases the inheritance can also occur without the transfer of the chromosome, which means the nuclear chromosome. So now we will be dealing the result of the dihybrid F2 cross. Okay, dihybrid F2 cross. Now we will be drawing the checkerboard. I hope you understand it. If you are having any doubt, you should mention it in the comment box. Okay. Now we are going to deal about the F2 cross. So what? And the I said. So how we will be getting F1? We will be getting. So how we will be getting F2? We will get F2 by selfing F1. So F1 selfing, which means F1 into F1. I mentioned that there are 4 types of gametes will be formed. What are the type of gamete? Capital R, capital Y, capital R, small r, capital Y, and small r, small y in parent 1. Okay, we are going to cross this in checkerboard. So this is parent 1 and parent 2. This is the condition. So I am going to, please write it. So I am going to draw the checkerboard right now. So how to draw a checkerboard? We know that here 4 types of gametes will be formed from the parent 1 and also parent 2. So 4 into 4, 16 types of progeny can be formed, right? So I will here mention 2 methods. One is the normal conventional method by which we draw a checkerboard, and next method is for the NEET, which means we will be drawing a shortcut method to write down the genotypes. Okay. So I'm going to draw a checkerboard. We will be getting 16. We need 16 columns. Sorry, 16 boxes, right? So we need to draw 4 columns and 4 rows also. So in a checkerboard, I hope this is clear. So 4 column, 1 2 3 4, 4 row, 1 2 3 and 4. So I am going to cross here. So here I will be writing any of the parent. I am taking sperm, which means male. Here I am taking the condition of egg, female. So capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. Okay. Here also capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. So I am doing a cross. So I think you understand what is a cross. So this gamete and this gamete can fuse. So I can write capital R, capital Y. This gamete fuse with here capital R, small y. So here 16 number of gametes will be formed. It is not actually type. I think I have said it as type, right? It is actually the number of gametes. We will see how much type will be formed, how much genotype will be formed. Okay, this is the checkerboard of we are dealing with sperm and also the egg from 2 different parents. So this was the condition of the F2. This much progeny was formed. Okay, this is the condition of the F2. Now we are going to analyze the result. We are going to analyze the phenotype. Okay, so I am going to write the phenotype here. Round yellow, round yellow, round yellow, round yellow, round yellow. So I think the majority is the round yellow. Here round green, round yellow. Here round green, round yellow. Here round yellow. Here wrinkled yellow. Here wrinkled yellow. I think you understand how I am writing it, because it is in the heterozygous dominant form. Round yellow, round green, wrinkled yellow, wrinkled green. So this is also the phenotypic condition. So this is actually a conventional method, the normal checkerboard drawing method. So I think you have written it. Now I am going to draw about how we can find a solution if it is given, if heterozygous gamete is given, and we need to find the F2 result. How we can write it? Here our F2 condition. Here our F1 heterozygous condition is capital R, small r, capital Y and small y. This is the F1 condition. So the shortcut is that I am going to draw, I am going to write a series of number alphabet, combination of alphabet. So we know that we have two gene, Y and R and Y gene. Okay, so first I will be writing capital R, capital R, capital R, capital R, capital R, capital R. Then it's heterozygous condition. Then it's homozygous recessive condition. 3 times. Okay, this is the condition of first gene. Now second gene. Homozygous, heterozygous, homozygous recessive. Okay, now we need to write number as 1:2:1. Then it's double 2:4:2. Then it's single 1:2:1. This is the method to write, shortcut method to write the genotypic and phenotypic ratio. Now we will be find out the phenotypic and genotypic ratio. Genotype and phenotypic ratio will be same, but we need to find the combination, right? So I will be writing the phenotype. Round yellow, round yellow, round green, round yellow, round yellow, round green, wrinkled yellow, wrinkled yellow, and wrinkled green, which is the genotype of the mother plant. So if we analyze the result, we can understand one thing, same as that of Mendel's dihybrid cross, monohybrid cross. One of the parent which was masked has been reappeared here, wrinkled, right? Reappearance, and also certain other combinations so have been also present, which is different that of the parental condition. It is because of the process of recombination, right? The parental condition different from that of the parental condition has also been observed. This was the parental condition, right? Okay, please write it down. Now we are going to analyze the result. So first I'm going to write. So round yellow, we are first looking on round yellow. So round yellow here 1, then 3, 5 plus 4, 9. So there are 9 round yellow type phenotype 9. Okay, then we are going to look about round green. Okay, round green here 1, then 3, then there is no round green, so round green 3. Okay, now we are going to look about wrinkled yellow. There is only 3 here, 1 and 2. So wrinkled yellow 3, and the last one is the wrinkled green, which is only 1 phenotype. This is the phenotypic ratio of the dihybrid cross. So we can understand many variety from the genotypic ratio. The genotypic ratio is 1:2:1 and 2:4:2, 1:2:1. This is the genotypic ratio of the dihybrid cross, and in case of the phenotypic ratio it is 9:3:3:1. So this is the shortcut method to find the genotypes. Okay, one thing to remember is that we can only use this shortcut method when the parent generation, that means the generation which we are selfing, F1 generation, should be in the heterozygous form. Okay, then only we can write this. So this is the case of Mendel's dihybrid cross. So from Mendel's dihybrid cross we were able to find out the law of independent assortment, which means the genes or the alleles of the gene segregate independently. Okay, this is the case of law of independent assortment. And I think we can do a question here. I will write the question. The question here is that I will say, determine the frequency of only one genotype. Determine the frequency of only one genotype, capital T, capital R, small r, in the offspring of the dihybrid parents. So there is a question given. The question is that what is the, or determine the frequency of the genotype. This is the genotype in the offspring of the dihybrid parents. The parent is this. This is the parent genotypic condition. So the question is that we need to find the frequency. Okay, frequency of this genotype combination. So what do you mean by frequency? Frequency, we know that the number of time a thing is happening, right? So we know that to find total number of times happening by total number. Okay, actually that is the frequency. We know that so this is the question, this is the parent. So we know that in frequency, how much number of time this genotypic combination occur, we need to find it. Okay, and we know that total number of occurrence is 16, right? We know that in a dihybrid cross it is 16, and it is also mentioned in the question, it is a dihybrid cross. And also we can find that two different characters are being taken, T and also R. Okay, T is a maybe it may codes for the tall height of the plant, and R for the shape of the seed, maybe. Okay, we don't know that, but it is the question is given that it is a dihybrid cross. So the parent generation is this condition. So we are going to find out the answer. So I am going to write the possible genotypic combination. We know that the parent generation is capital T, small t, capital R, small r. So how to write? The shortcut form is that we need to write capital T. This is the way. Okay, I earlier mentioned. Next, capital R. Okay, this is how we write, and we know that genotypic ratio is 1:2:1, 2:4:2, 1:2:1. So we need to find from this possible number, we need to find this combination, right? This is the combination. Okay, we can only get this combination, rest of them is not applicable. Okay, so out of 16 progeny, 2 progeny are formed. So 2 by 16 or 1 by 8. Okay, so this is the answer, 1 by 8. Yeah. So this is the frequency, number of time occurring. Okay, so we will write. So we will discuss the next. We will discuss question while we are discussing the entire previous question paper of NEET examination. Okay. From this, so this is the end of the lecture 3, in which we have only dealt with the dihybrid cross and also Mendel's law of independent assortment. From the previous 3 lecture, we first deal with the basics. The second was monohybrid cross, law of independent, law of segregation, law of dominance, and also multiple allelism. Now we have dealt with the Mendel's dihybrid cross and also law of independent assortment. In the next lecture, we will be dealing about, here we have also dealt with chromosomal basis of inheritance, which means during the Mendel's time, while he was doing the experiment, he has just concluded the experiments on the basis of the phenotypic analysis, right? Just analyzing the phenotype. But later, 50 years later, there was he got a basis. His experiment got a base. The base was the chromosomal basis of inheritance. So there was a strong evidence or strong proof that Mendel's cross is true. Okay, so this was the case of Mendel's dihybrid cross. So this is the end of the lecture 3, and in the next lecture we will deal with polygenic inheritance and sex determination, mutation, pleiotropy, etc., which are very small portions, and there is also another portion which is the linkage and recombinants, which is also an important. So I hope you like my lecture. Thank you.